what I want to do today is I want to talk about autism, start from experiences that perhaps many of you have had with autistic people who you've met, and then I want to turn to the biology of it and use, actually the recent piece I had in the Times, use risk ratios to help frame how autism looks to someone who's not a scientist and things you may have heard about in the mass media. I want to then take those risk ratios, which I'm going to use to illustrate what you hear about in the media, and I'm going to flip it around and I'm going to show you what it looks like to me as a working scientist. And so I want to turn it around and show you the same thing, looking again at those same risk ratios. And then I want to give you a little insight into what some researchers in the field are thinking about now as to what causes autism. So that's roughly the trajectory of where I'd like to get to. Now, at the risk of telling you something that you already know, let me just talk about autism a little tiny bit. To oversimplify and to just get down to the nut of what people think of as autism. Think of autism as a disorder now diagnosed at well over 1 in 100 small children in the US population. Think of that autism as a disorder of aloneness and sameness. So relative to other children who the autism community refers to as neurotypical, autistic kids have difficulties with social interaction, they have difficulties with language, with give and take with other people, and in some fundamental way there's something very alone about autistic kids. Another thing that characterizes autistic kids is that they crave sameness, that perseveration where they like one thing over and over again. And often parents will notice, one of the first things they'll notice in their kids is maybe the kid will freak out when, will become very upset when taken to daycare by a different route or by taken on a walk by a different route. And that love of sameness is something that appears very early in life. Now, I realize that I've put together a lot of different things that come under the umbrella of autism spectrum disorder with this very simplified definition. And I should say at the outset that if you've ever met more than one person of autism, there's a saying in the community which is that if you've met one person with autism, then you have met one person with autism. And so there's a very wide range of phenotypes, and that's why it's often referred to as autism spectrum disorder. Before getting into the biology of autism, I want to talk a little bit more broadly about general development, general brain development. Because it turns out, as far as I can tell from looking at the literature of normal brain development, which I've studied for a long time in my laboratory, and normal brain plasticity on the one hand, and autism on the other hand, there are a lot of commonalities as to what leads to a typical neural path of development as opposed to going down the path of autism spectrum disorder. And a lot of principles come up over and over again. And so the first thing I want to introduce in the few slides that I want to show is the idea that there's an interplay between genes and experience and environment, and that is the case whether we are not on the spectrum or whether we are on the spectrum. And so this is just a slide to remind you about a lot of things that we care about in ourselves and perhaps our loved ones, whoever they are. And there are a lot of things that we think about as defining us as individuals, things like personality, memory, identity, normal function that we experience in everyday life, and also dysfunction, things like autism. Now, over here on the left are the themes that I want to talk about here, and these themes are the same, whether for neurotypical people as well as people who are on the spectrum. The idea that genes give us a starting point. Those genes are a program that we get when we're conceived. Then that works through developmental programs, developmental mechanisms, and then the environment and experience then shape that genetic program to make us who we are and make us all these things on the right. And that story is the same for everyone, whether on the spectrum or not. So I want to use that as a common starting point. And I want to talk just a little bit about typical development and also autistic development, and just point out some pretty remarkable things about child development. I should say that I'm a parent, and when my wife and I discovered that we were expecting a baby, I did what any good academic does, which is that I waited till the last minute and I started cramming. And I went to the bookstore and I started reading books about child development. And I discovered these books like, if you know these books at all, it's things like Healthy Sleep Habits, Healthy Child, What to Expect When You're Expecting, right? The Happiest Kid on the Block. And if you stop and think about all those books, or nearly all of them, touch in one way or another on brain function. But yet these books didn't really cover brain mechanisms at all. And so I started reading about that. And so while we were thinking about our new baby, and who is due to arrive any moment, I also started discovering some pretty amazing things. From birth to about age 1, there is this incredible proliferation of synapses. So if you see a baby in front of you, that baby is cute, is gurgling, spitting up. That baby is forming tens of millions of synapses per second. And that is just this cauldron of activity, a cauldron of birth of synapses, which then crests at about age 1. And then around age 1, synapses start disappearing, which is somewhat surprising. I was actually pretty surprised when I learned this. And synapses begin to disappear at a net rate of tens of millions of connections per second. And that is a somewhat arresting thought, because naively, perhaps we think about synapses as being good, right? We joke about losing synapses, whether we drink alcohol or we get older. We think about synapses as being good for function. But here, we have something that's quite curious about child brain development, which is that children are in the business of getting rid of synapses. And what that means is that there's some kind of shaping, some kind of pruning of brain architecture that's happening in all children. And so if you think about children, all children that you meet, yourself when you were a child, think about that formation of synapses and then disappearing synapses. It's this crazy pruning activity that involves biological mechanisms working to form connections and then in some sense to get rid of the connections that are not needed, right? A pruning activity during what neuroscientists call a sensitive period of development, where there's this proliferation of dendrites to form many synapses, and then the ones that are not needed get taken away. And so really, think about the growing brain as a physical organ that shapes itself by what developmental biologists call progressive events, where there's growth, and regressive events, where there's retraction or loss of synapses. Now, so far, everything I've told you is about all brains of all children. But now let me just switch to autism a little bit and talk about some curious findings in autistic children and use that as a place to start thinking about autism and what's going on. So this is a paper from Eric Courchain's lab in California, in San Diego, I believe. And this is a graph now of brain volume on the vertical axis and age. And blue is neurotypical. And you can see here that typical kids have a rise in brain volume that then peaks and then levels off and perhaps even goes down a little bit. But if you look at kids with an autism diagnosis, there are a couple of striking things going on here. One is that they have actually larger brains, and actually it's been observed for a long time that they have slightly larger heads. And so they have slightly larger brains, and furthermore, there's this lack of cresting here. You can see this red average line of all these measurements. It seems to be going up and doesn't really quite crest. And so it seems like there's something different going on, even from a very early age, in the brains of autistic kids. And I think that one way to think about this is to think about the pruning that I was talking about before, that pruning that happens so normally in so many brains, in most kids, may not be happening in exactly the same way. There's something where that pruning is not quite happening in the same way, perhaps not happening quite as well. So I want to plant that idea with you, and I want to come back to it in the rest of the slides, a few slides later, and perhaps during discussion. Let me just switch gears a little bit and talk about risks for autism. And some of you may have seen my piece in the Times, and let me just talk about risks for autism. So we hear about many things in the news about what may or may not cause autism. For instance, this is a pretty savvy audience, and so I don't need to talk about vaccines too much except to say that they don't cause autism. But let me just talk about all the risk factors that you may have heard about, whether vaccines, maternal age, right? That's always a good one to make people feel guilty. Honey, you're not married yet. Do you have any idea what you're doing? I saw an article in the paper. If you're 35 and you haven't had a baby yet, your kid is going to meet a nice boy. So I want to take these risk factors and put them into perspective. And if we think about the rate of autism in the general population, let's call that 1.0x, right? That's the general rate as measured by the Centers for Disease Control or in any population. That's 1x. Obviously, the reported rate of autism in children who have gotten the MMR vaccine is also 1.0x. So there's no difference in the rate of autism in babies who have been vaccinated versus babies who have not. Let me now fill in this slide a little bit. This maternal age thing that you've heard about, mom over the age of 35, that's a 1.3-fold increase. So if the rate is 1 in 100, then that risk is now 1.3 out of 100. Dad over the age of 40, 1.4x. Okay, so these are the risks that we hear about, and if your attitude is that no risk is too small, then you would be concerned about this, and I wouldn't necessarily fault you for that. But I want to fill this in and give you a biological picture of some other risks that can teach us a little bit about the biology of autism, a little bit. And at least for me as a working scientist, they've made me think a little bit differently about the causes of autism. Elective cesarean, right? Cesarean when not medically recommended. It used to happen for about 1 in 12 deliveries in the US. That rate has gone down in the last few years, actually dramatically. Increase in risk is 80%, 1.8-fold. Emigrating from one's home country while pregnant more than doubles the risk of autism. Pregnant mom caught in a hurricane strike zone triples the risk of autism, and that's if the mother gets caught in a hurricane strike zone during the second half of pregnancy. Now, there's something going on here, and I want to tie these together, and I want to talk about this a little bit. You know what? In the interest of time, I'll give away the ending: stress. Okay, so this is probably, at least in part, mediated by stress hormones, which are helpful in the short term, but on a persistence basis can damage organs and can also damage the brain. And so one possible way to think about these different factors is stress. But let's just keep on going a little bit. Premature, more than 6 weeks, that's a big one. There are these famous cases of babies who are put up in orphanages in Romania, and there are certain types of orphanages where babies get no loving care, no touch, no social attention, and there's really a pretty remarkable increase in the risk of autism in those babies. But now, so far, I've already shown you some risks that are greater than these ones that you hear about a lot, and here are the ones that I want to really focus on for the remaining few minutes. Sharing half your DNA, fraternal twin, cerebellar damage, identical twin. Okay, if you share all your DNA, or nearly all your DNA, with somebody with autism, the risk is increased over 80-fold, right? These are, I hope you will see that these are much larger than these risks over here. And so meanwhile, while we read in the news about these risk factors down here, there is something else going on biologically, and I want to talk about that a little bit. And so I want to just spend the next few slides talking about fraternal twins and identical twins, and that's roughly speaking genetic inheritance. And then I want to talk about this other thing, cerebellar damage, which is, this is a brain region that I study, and I've studied even before I became interested in autism, where the risk of scoring high on autism inventories is increased tremendously. So there's something else going on here, and I'll focus on these red bars for the remainder of the slides. So the picture I want you to get out of this whole set of bars here is that predominantly, more than anything else, think about autism as a genetic disorder where combinations of genes lead to increased risk for autism. Think of it as possibly a situation in which prenatal factors, environmental factors that we haven't necessarily identified yet, some kind of environmental factors increase the risk for autism. One possibility, as I've mentioned, is stress hormones. Another possibility is illness. This is very much an open question. And finally, the possibility that I want to end with tonight, which is the possibility that abnormal input to the cerebral cortex, the 76% of our brain that distinguishes us from other mammals, that maybe abnormal input to the cerebral cortex may be at the root of autism. And that's going to be what I talk about in regard to cerebellar damage. Why do we have these genes? You can see risk genes, genes that increase the risk of autism, all over the genome. What are these doing here? Turns out that any one gene only has a weak effect, only increases the risk of autism modestly. So we've got hundreds of genes that have been identified, each of which, most of which increase the risk of autism modestly. A few of them increase the risk a lot. So what's going on? Why are we carrying these genes? Presumably by something like, say, natural selection, we would get rid of these genes. It's not necessarily the case that it's not immediately obvious why we would be carrying these genes. So I want to talk about that a little bit. This is now, I'm going to show you the results of a survey I did of Princeton undergraduates. They just graduated, but when they came in as freshmen, I had the honor of addressing them giving them their first lecture, and I said, "Welcome to Princeton, I'll be speaking to you in September. By the way, could you fill out this survey?" It turns out that newly admitted freshmen will do anything a Princeton professor asks them. So I got a 90% response rate. This is fantastic. 90% response rate on a survey. It was amazing. So I asked them, their attitudes about science, I asked them what they might major in, and I asked them whether they had relatives, first-degree relatives with neuropsychiatric disorders and neurodevelopmental disorders, whether it be bipolar, mood disorder, substance abuse, Parkinson's, autism. And let me show you one of the things that I found out. So the humanities and social sciences majors had a rate of autism in their siblings that was similar to the general population. And on average, they had more than one sibling. So this is not any different from the general population. But look what else I found. Science, math, and engineering majors had a rate of autism in their first-degree relatives of more than 3 times their humanist and social science classmates, right? A more than 3-fold increased rate. So this is pretty interesting. And I should just interject at this point that in college I was a physics major, as you heard, and I should mention in full disclosure that my sister is autistic. So in fact, I am that little green guy right there, and perhaps some of you are as well. So what's going on here? Well, an idea that we're really excited about finding out more about, and we're starting to investigate in my laboratory using modern tools of genomics, is to find out whether there are genes that do one thing in some kids, that is, send them down the road to autism, and in other kids, send them down the road towards being interested in science. And so we're super interested in that because if these genes only have small effects, that means that many of you are carrying around these risk genes. And they're clearly not putting you on the spectrum. So what are they doing instead? Well, I mentioned that genetics is the starting point, right? And without getting too much into it, so many aspects of our individuality have now demonstrated to be inherited in some way. Personality, verbal ability, mathematical ability, neuropsychiatric disorders, all of the ones I listed. So I want to just give you a very brief quiz, and I want you to think about this a little bit. So for those of you who have college degrees, I want you to think about, A, your college degree, but B, I want you to assign yourself a score. Think about all your first-degree relatives. If you have a sibling or a parent or a child with autism, give yourself a score of -1. If you have depression in your siblings, parents, or children, +1. Bipolar, +1. Substance abuse, +1. All of these are heritable disorders. And this is a plot of what we discovered in Princeton undergraduates, which is if they scored -1, so they had autism but none of these 3 other things, then they were overwhelmingly likely to aspire to natural science or engineering majors. At the other end, if they scored 2, which is 2 of these 3, or perhaps all 4 of these things in their families, which would be quite a family, then at that end, then that's 2 to 1 in favor of being a humanist or a social science major. So I hope by now I've given you time to add up that score and think about that, and think about adding up that score and seeing which of these colors you are in here and see whether you fit. And I think that statistically most of you will find that it's not too contradictory to your everyday experience. So the idea here then, as I said, is that genes can lead us down many developmental paths, in some cases making us individual in different ways, in other cases leading to neuropsychiatric disorders, which in some ways is an extreme form of individuality. And the idea that we're pursuing right now is that those same genes have both jobs. And so perhaps the job of these genes is to create diversity in us intellectually. Let me talk about some other things that we're doing. So it turns out that we're now working with the Simons Foundation, which has a thing called the Simplex Collection, which is families with exactly one autistic child. And what they've done is they've gathered a whole lot of phenotype information, giving surveys, taking blood samples, tissue samples, and so on. And one of the things they've done is administered this thing called the Broad Autism Phenotype Questionnaire. And we got very interested in this because it's our first attempt to start doing genetics on individual personality. And I'm going to read to you some of these survey questions. So I took the survey and I got really interested in it. I like being around other people. Very rarely 1 to very often 6. I would rather talk to people to get information than to socialize. Well, duh, of course. I am flexible about how things should be done. I've been told that I talk too much about certain topics. I can tell when someone is not interested in what I am saying. So at this point in the survey, I started flushing a hot red because I just felt myself answering yes to a lot of these questions. Did I mention that I share half my DNA with an autistic person? So the interesting thing here is that if you score high enough in these domains, which are in things like aloofness, communication, that the prevalence of this phenotype, this broad autism phenotype, which is not quite autism, but it's a set of personality characteristics that are reminiscent of autism. In the general population, you can find these characteristics in 5 to 9% of parents. In parents of kids with autism who share half their DNA with the autistic kids, that rate is 14 to 23%. So this is a big tell. This says there is something in common between members in that family. Could be environmental, could be genetic. And we are super interested in that. And what we've already found is we've started to find some hits that are statistically significant or near significant, where there are genes that, variants of genes that seem to increase the likelihood of this phenotype and also are known risk factors for autism. And so we're very interested in that. So our hypothesis is that shared genes govern intellectual interests, personality, and neuropsychiatric problems. Now let me just switch one more time before the end and talk about biological mechanisms. So far I've talked about genes, and if any of you is familiar with genetic research, there is a long road between discovering genes and understanding at some systems level what is going on in a developing brain, what's going on in terms of a circuit, whether it be autism or schizophrenia or Alzheimer's, what happens functionally. If we start talking about big brain structures like hippocampus or cerebellum or amygdala, it's a long, long road of explanation from genes to really having some kind of organic synthetic explanation of what's going on. One can detect sensory differences between autistic kids and typically developing kids as early as 4 months of age. They take a little longer to respond to an auditory stimulus. And by age 2, they have trouble associating visual biological motion from the sound of biological motion. So what's going on here? Well, the thing that we're really interested in is the possibility that sensory experience is necessary for building a social brain. Remember I told you about the Romanian kids, those poor kids who were put in an orphanage and they get no social experience whatsoever. They're isolated from experience. You may have heard about Harlow's monkeys, where the thing with the wire mom and the cloth mom, and then the monkey goes for the cloth mom. So one thing that is not said about those monkeys is those monkeys are raised without any kind of biological mother. And one thing that the monkeys do after being raised by wire or cloth mom, either way, is they start doing things like this, right? This is an autistic move. They start self-stimming. They don't behave appropriately. And so sensory isolation, as horrific as it sounds, either in Romania or with Harlow's monkeys, appears to be a way to send brains down a path that looks a lot like autism. So what if there was something in the brain that interpreted sensory experience and softened it up and helped the rest of our brain understand it, right? There's nothing intrinsic about an arc of light appearing through the trees that would make you think that it was, say, your mother smiling at you or just light streaming down from the sun. There's nothing intrinsic about that. We learn from our environments how to be social. So what if the brain needs internal brain mechanisms to learn to be social? And this is what we're working on right now, cerebellar damage, right? This cerebellum, this structure that's present in us and also in rodents, where my laboratory mainly studies it, this structure here is important for processing sensory experience. For a long time, people have been interested in the cerebellum as a structure that helps with balance and movement. But we're very interested in the idea that the cerebellum also may be a developmental teacher early in life. And the evidence for this is that we know that the sensory job of the cerebellum. The cerebellum is important for processing sensory information. For instance, the cerebellum is activated differently when we go like this to ourselves or when somebody else does it, right, like with a feather. So the cerebellum is thought to play a role in why it is that we can't tickle ourselves. You should try that. That basically the cerebellum is important for detecting discrepancies in the sensory environment. And so it's been long known that the cerebellum is somewhere between sensation and action. So we're interested in a new idea, which is that the cerebellum also might be intermediate between sensory experience and social development. And so we're really interested in the idea that the cerebellum participates in shaping the social neocortex, parts of the brain that are important for social engagement during periods of development where the ground is nice and soft, metaphorically speaking, where development is capable of changing the brain. And this is the idea that we're testing. There are connections between the cerebellum and the neocortex. I won't get into this because I'm a little short on time, but suffice it to say that there are loops of connectivity between the cerebellum and the neocortex, and those loops go in both directions, so the connections are there to play the teaching role. And I left out the slide, but it's been demonstrated that when mice have a gene that's deleted only in cerebellar neurons, those mice develop symptoms that look a lot like autism, where the mouse version of autism is being indifferent to other mice versus inanimate objects, not vocalizing properly, or repetitive self-grooming, right? So mice don't have language, but there are other things that we can see in mice that seem kind of like a mouse-like version of autism. So the ideas that we're very excited about right now in my laboratory are, we know from work in our laboratory and others that the cerebellum is a multisensory processor for processing different sensations that come in from the world. It's been observed clinically, and also I told you in rodents, that cerebellar function and dysfunction in early life leads to autism in humans and something that looks like autism in mice. And so we're super interested in the idea that these risk factors, these genetic risk factors I've been telling you about, might lead to early life difficulties in sensory processing. And we know that experience is a guide for social brain development. And so the idea that we want to test in the next few years is the possibility that dysfunctions in sensory processing could lead to autism. And we're doing this in rodents, as I said. This is now the cerebellum of an anesthetized rodent expressing green fluorescent protein. It's actually a version of green fluorescent protein that is sensitive to neural activity. So these neurons get brighter when they are active. And we can look at this in the living brain of an animal. And you can see here that these cerebellar neurons are quite beautiful to look at in vivo. At least they're beautiful to me anyway. And so we're very interested in looking at this activity and perhaps seeing what role it has in teaching the social brain. I will stop there, and I would be delighted to have a conversation with you all now. Thank you.